So the last and final step of photosynthesis, coming after the light-dependent reactions, is the Calvin cycle. And remember that the Calvin cycle was what provided us with energy for, the light-dependent reactions is what provided us with energy for the Calvin cycle to happen. So that's where we got our ATP and NADPH in order to then turn glucose into carbon dioxide and sugar. So the energy for what we're going to talk about is coming from the light-dependent reactions. And this is a cycle. So that means that it regenerates the starting material after molecules enter and leave the cycle. So like in the citric acid cycle, oxaloacetate accepted acetyl-CoA. In this case, the molecule that accepts whatever enters the cycle is going to be regenerated and used again to again accept whatever is going to be entering the cycle. And the cycle builds small sugars. So we're going to see what that means, but we use ATP and NADPH, or the electrons carried by NADPH, to power this process. So carbon enters the citric acid cycle as carbon dioxide and leaves as a sugar named glyceraldehyde 3-phosphate, which we're going to call G3P from here on out. Notice that we're not saying glucose. It does not leave the cycle as glucose. It's really easy, especially in lower biology, to say it leaves as glucose, but actually it leaves as G3P, which can then later be built into glucose or other sugar molecules. And so to make one G3P from the Calvin cycle, it needs to happen 3 times. So that means that 3 times 3 different molecules of CO2 need to come in. So the Calvin cycle has 3 phases, which you'll need to know. Carbon fixation, which is Rubisco, which you will know what that means. Carbon reduction, and regeneration of the CO2 acceptor, which is RUBP. So there's 3 different stages. The first step, which we see right here, is carbon fixation. What this means, do I have a slide for this? Nope, okay. So this is carbon fixation. Fixation means, it's the initial reincorporation of a carbon dioxide into an organic material. So carbon in carbon dioxide is not technically an organic compound. Remember, it needs to have carbon and hydrogen. Carbon dioxide is none of these. It's not a lipid, macromolecule, protein, or nucleic acid. It's not any of them. So we need to fix it into an organic molecule. That means take the carbon and make a carbon-carbon bond with another organic molecule. And so that's what happens in carbon fixation. So in this cycle, you do actually need to know a couple of things that happen. First and foremost, we have what's this acceptor, which is what's going to accept the carbon dioxide, and it's called ribulose bisphosphate, or RUBP. It's made of 5 carbons. We're going to count carbons again, just like we did in photosynthesis. Cellular respiration. So there's 5 carbons. RUBP has 5 carbons. Carbon dioxide comes along, and then it has 6, because this carbon dioxide gets fixed to this one, and now it's a 6-carbon sugar. The enzyme that attaches carbon dioxide to RUBP is what is called Rubisco, and Rubisco stands for RUBP carboxylase, because we're adding the carbon to RUBP. So what it comes from is ribulose bisphosphate. So it's never mind, I don't know what it stands for. But Rubisco is the enzyme that attaches carbon dioxide to RUBP during carbon fixation. It is definitely the most abundant enzyme inside a chloroplast, and probably the most abundant enzyme in all of Earth, because that's how important this step right here is, because every living thing uses the products from photosynthesis. So after this gets fixed to RUBP via Rubisco, it splits in a second. The 6-carbon molecule is very unstable, so it immediately becomes 6 3-carbon molecules. Now let's count our carbons. We started off with a 5-carbon molecule right here, but remember this is happening 3 times. So the counting is very tricky. We have 3 5-carbon molecules. That's 15 carbons. Then we add 3 carbon dioxides. So now we have 18 carbons. That's how many carbons we have right now. At first, they're in the form of 3 6-carbon molecules, still 18 carbons. Then they all split in half, so now we have 6 3-carbon molecules, still 16 carbons, or 18 carbons. So that's carbon fixation, when we attach a carbon dioxide to RUBP via Rubisco. Then, all the carbons that we have, all 18 of them, get reduced into G3P. You don't need to know the names of these molecules or anything, but basically we had this 3-carbon molecule, and we're going to reduce it to G3P, glyceraldehyde 3-phosphate. To do so, we need to put in 6 ATP and 6 NADPH. So this is where we're providing energy to change something. That energy was made in the light-dependent reactions and now is being used to reduce a carbon molecule to G3P. So at the end of the carbon reduction stage, we have 6 G3P molecules, okay? Still 18. Now, 5 of those are going to go back into the Calvin cycle. 1 of them is going to leave as G3P, and that's the product. So this is where the product leaves. The product is G3P, and it leaves to go become glucose or other organic molecules. Okay, so this is the product right here. G3P is the product of the Calvin cycle. You can see there's 3 sugars, so that's 3, and 5 G3Ps are staying in the cycle to go back. So before this G3P leaves, we still had 18 carbons, and once it leaves, we have 15 carbons, which remember is exactly how many that we started with. So we have the right amount of carbons, but now we have to change them back into the form that they started with, because right here they are G3P, but up here it was a 5-carbon molecule. So we need to do some reformation. So here we have 5 G3P molecules, so we have 5 3-carbon molecules, and those get regenerated back into RUBP. So here we have 15 carbons, 5 3-carbon molecules. Here we have 15 carbons in 3 5-carbon molecules. So this step is called regeneration of the CO2 acceptor. So we're regenerating RUBP, and it also requires some ATP to turn these 5 G3P molecules into 3 RUBP molecules. And so these steps right here that require ATP is why cyclic electron flow happens sometimes in the light-dependent reactions. Think back to that. Because overall we need 9 ATP for the Calvin cycle and only 6 NADH. And the light-dependent reactions, when they are using linear electron flow, make the same number of NADH and ATP. So we're going to need somehow to come up with extra ATP, otherwise we're always going to be short on ATP and have too much NADH. So to do so, we use cyclic electron flow. So the 3 steps of the Calvin cycle are carbon fixation, where 3 carbon dioxides come in, they get fixed to 3 different RUBPs from Rubisco. The end of carbon fixation, we have 6 3-carbon molecules. We have 18 carbon molecules altogether. Then those 6 3-carbon molecules get reduced through ATP and NADH to 6 G3Ps. This is the final product of the Calvin cycle. So we have 18, 3 of those leave as 1 G3P, and then we have 5 G3Ps left over. Those G3Ps get regenerated into 3 RUBPs with the help of 3 ATP during regeneration of the CO2 acceptor. And that is what you need to know about the Calvin cycle.